Hello, my name is Ralston Smith, and I'm a student in the Department of Chemistry and Biochemistry at GMU. The title of my project is the development of a novel Granzyme B PET imaging agent for the assessment of cancer immunotherapy efficacy. My mentor is Dr. Solomon. I am developing a Granzyme B binding peptide that is ultimately intended to be used non-invasively in the assessment of cancer immunotherapy efficacy through radioactive imaging. Let's begin with some definitions. A peptide is a string of amino acids, and amino acids are the building blocks of proteins. Granzyme B is a protease, which is a protein that cuts proteins, and it has important functions in programmed cell death, or apoptosis of cancer cells. This is a computational model of Granzyme B. Finally, efficacy is the propensity of a treatment to have the intended effects for an individual. One of the main issues with cancer immunotherapy is determining efficacy, and those who respond poorly to treatment have a high likelihood to experience severe adverse events, including death. Reducing this difficulty in efficacy assessment is what this project is about. The mediation of programmed cell death by Granzyme B is important because elevated tumoral presence of Granzyme B indicates a potential positive responder to immunotherapy. By developing an imaging agent that binds Granzyme B with high affinity, we seek to monitor these conditions to determine efficacy. This semester, I have been working in the lab with these peptide variants that I computationally identified as the best Granzyme B binders. As you can see, all the variants have IEXD sequences, meaning that each variant is comprised of a conserved isoleucine, glutamine, and aspartate in the first, second, and fourth positions respectively, with a variable amino acid in the third position. We placed an order for these 5 of the peptide variants to study this semester. To determine the variant with the highest affinity, I am performing a series of surface plasmon resonance or SPR experiments. This technique measures changes in light diffraction to characterize binding. I have recently collected control data from the 5 peptide variants we purchased to see how they interact with the instrument in the absence of Granzyme B. First, the very strong signal to the far left is the response to a bubble in the system. This signal can be ignored. These small signals here and here are representative of the peptide variants interacting with the instrument. These strong signals here represent imidazole washes and nickel injections, not peptide samples, so they can be ignored too. The control data overall is looking promising because there is little to no observed signal, meaning the signal that we will obtain during the enzymatic trials should be accurate with minimal noise. I will be continuing my work on this project over the summer, and I'm excited to gather more data moving forward. I would like to thank Dr. Solomon for his ongoing support and leadership on this project. Thank you to Dr. Foreman for sharing his expertise in computational data collection, and Dr. Veneziano for providing access to his SPR instrument. Thank you to Robert Spain, Alison Snodderly, and Yuande Oyasola for their extensive laboratory training and advice. Thank you to Zach Bulak for helping to confirm the purity and identity of our peptides. A special thank you to Dr. Ikutan, Assistant Professor in the Department of Molecular and Medical Pharmacology at UCLA, for inviting us to work on this project. Finally, I would like to thank Dr. Lee and the Oscar URSP for funding this project. These are my references. Thank you for listening.